Hello, good afternoon. My name is Alla Karnovsky, and I am going to talk to you about some pathway analysis tools. And so I have two talks, one before the break, and then we'll have a short break, and then we'll get to the second part. So the plan is to, in the first part, what I would like to do, I would like to, like I said, to talk about pathway mapping, and in the second part, I would like to talk to you about limitations of the pathway mapping techniques and the newer tools that hopefully you will see the how you can apply this to your own data. And also, I'm going to give you a little bit of a preview of a hands-on demo that I know many of you signed up for tomorrow, so you will know what to expect to see tomorrow. So, well, if you haven't seen this enough by now, you're looking at this again, and we are sort of at the end of the at the end of this journey. And as much as you might enjoy the journey, the destination really is, and the quest is to not just identify these metabolites and these changes, but to try to understand what it really means in terms of the mechanism of the biological phenomena or disease that you're studying. And so, as I said, the second part of the talk will be devoted about talking about what we can do beyond and how we can go beyond the biochemical pathways. So we have spent some time, George described to you various statistical analysis techniques, and so what really sort of at the end of the day, what comes out of this is some kind of a ranked list of metabolites. And this is typically a starting point for the pathway mapping and the enrichment analysis that George described. And so the idea behind the pathway mapping is really simple, and it seems to be very intuitive, right? So we do know a lot about metabolism, at least primary metabolism, and all these metabolic pathways, they are out there. And some of us, I mean, if you're like Chuck, then you have them all in your head. If for the rest of us, there are biochemistry books and there are databases and so on and so forth. So anyway, but I mean, it seems like intuitively, it seems like there are a lot of known relationships. We know a lot about enzymes. We know a lot about biochemical reactions and pathways. And it seems like this information should be handy in really trying to understand mapping your metabolites. And you have seen throughout the workshop, you have seen some examples of that, where these different metabolites are and what these different changes and different pathways really mean. Okay, and so there are lots of databases, including pathway databases out there, and so any Google search will, return a lot of information. And I kind of wanted to, and a number of different databases were also mentioned here. So this is meant to kind of help you get oriented within this sort of database world that is out there. And so I think, I mean, there's no really good way. I mean, a lot of these databases is fairly comprehensive. They cover compounds, they cover reactions and pathways and enzymes and genes and so on and so forth. There are databases that are more chemistry oriented, that have compounds and spectra. So this is sort of, roughly grouping them. So Maureen in particular mentioned, I think, all of these in her talk, and many of these databases, such as Metlin, can be used for compound identification. By the way, this is sort of a black box and meant to be a black box, and the pathway mapping is somewhat less of a black box. It's kind of a gray box. I will try to kind of, clarify what goes within that box. And some of these databases also contain spectral data, which helps, again, with compound identification and gives you a lot of information. Another newer category of the databases, if you will, which we have not talked about, but which you will hear quite a bit tomorrow afternoon, I believe, are the repositories for the experimental data. And so in particular, I want to mention the metabolomics workbench, which is being developed by the group at UC San Diego. And there are our collaborators and partners. And so the NIH mandate for our center is, in the other NIH-funded centers, is to deposit the experimental data. And so if you are funded by NIH, and if you have a pilot, or if you have a pilot grant with us, you're actually obligated to deposit your data to Metabolomics Workbench. Another database here, by the way, I mean, you guys have the slides, and all these are hyperlinks, so you have all the information if you want to look at these. Metabolites is sort of a European equivalent of Metabolomics Workbench that is being developed by at EBI, European Bioinformatics Institute. And many of these come, I mean, both of these come with their own set of tools for compound identification, for doing some basic statistics, and so on and so forth. But the type of databases are sort of, primary relevance to my talk are the pathway databases. And so the first of this is KEG, which many of you, I'm sure, know. It stands for Kyoto Encyclopedia of Genes and Genomes from Japan. The first database of its kind. It's a pretty comprehensive database of biological pathways, and that also includes metabolic pathways, and the other databases, I'll just sort of mention them. And so the reason it's important to talk about this is because, I mean, there are many tools for doing pathway mappings, pathway mapping including some commercial tools. And with these tools, I mean, you often, and I often heard complain, and I have complained about this myself, yeah, I'm doing something, but there's really a black box, and I don't know what is going on inside that black box. How is that pathway mapping happening? Okay, so the tools that I'm going to primarily talk to you about today, I will be able to explain to you how we do the mapping, but again, sort of, briefly to talk about KEG, it covers, variety of biological pathways. And I think in the interest of time, I can sort of, go directly. I have a couple of examples here, a signaling pathway and a metabolic pathway, glycolysis. So the thing to know about KEG database is that if you want to go and query this database and look at the pathway charts, you can do that. If you try to download the data, you need a license to do that. So it's free for sort of, everyday use and doing sort of, searches on the web. They also have API if you're interested in more sort of, programmatic access. Again, be careful because access to the data is sort of limited, and they don't allow to redistribute the data. The next database, or a family of databases, if you will, called Psyche, would be probably of primary interest to those of you who are interested in bacterial metabolomes, who may be working in the microbiome and related subjects, because this database is known. I mean, one of the databases contained in Psyche, and perhaps the best known, is called EcoPsych for E. coli, and it is very well curated and very detailed database. In addition to EcoPsych, they sort of, they have many different organisms in there, and they divide their databases into 3 tiers depending on the level of detail of their annotations. And so tier 1 databases are sort of, manually curated and, require a lot of maintenance. Tier 2 databases undergo some manual review, and tier 3 databases are generated in a completely automated fashion. But still, if your microorganism, if that's only data about pathways in your particular organism that are out there, it still can be handy. Okay, so so you already know that I will be talking about Netscape, the tool that we developed here, and fortunately for us, when we started, at that time, some really detailed and really high quality databases were published for human metabolic pathways. And this include Edinburgh Human Metabolic Networks and database from another group that is called Recon. At the time, it was Recon 1. And so what's special about these databases? So you can, somewhere in my slides, right here in the title, I have metabolic reconstructions. It's important to understand how these are created. So essentially, to create this, what is called metabolic reconstruction, they start with a set of genes in a given organism, in this case, a set of human genes. And then, from this, identify all known metabolic enzymes, and then essentially create a catalog of metabolic reactions. And that's how they go about reconstructing all these pathways. And so literature curation is an essential part of that. And so these are essentially very highly sort of curated, carefully designed databases. And it's important because it reflects the quality of data. Is there a question? Sorry. Okay. And so essentially the reasoning for doing this pathway mapping, all the reason is represented here. So as I said before, you want to go from a list of metabolites to a map. So here you have your significant metabolites, and you want to be able to sort of, map them into here. And so the other sort of aspect of this pathway mapping that I want to emphasize is that I think you have seen by now that the data sets that you're going to get is going to be incredibly complex, multi-dimensional data. And the general rule with this type of data is the more ways you have that, the more ways you find to look at your data, the better off you are, okay? The more things you're going to see. If you're doing a very simple experiment, you have a, 1 table, 2 tables, maybe you're fine. Here, essentially, I guess the message I want to convey is that visualization, in this case, is a form of data analysis. Okay, so looking at your data is, there's nothing that can substitute that. Okay, and so that's sort of, the reason and the motivation for developing, multiple sort of, pathway mapping tools and multiple visualizations. And you have seen this slide, I think, before, so I won't, spend too much time talking about this. But again, there are some links out here that you can use, and you can go and explore these tools on your own. So I think in the interest of time, I'll just sort of, briefly mention 2 commercial tools. Many of you may be familiar with them. One is Ingenuity pathway analysis tools, and they handle metabolomics data and they handle gene expression data. It's pricey, but you can get a free trial version and at least get an idea of what you can do with that. Another one is from the company that used to be called GeneGo. Now they're part of Thomson Reuters. And nonetheless, I mean, it's a useful tool. I mean, it has advantages and disadvantages. So, you know, I'm not associated in any way with either of these companies. So I'll just sort of, you know, stay neutral. And just know that this is a possible option for analyzing your data, especially if your institution has access to this. I mean, the more tools you try, the better off you are. Okay, so I think I've touched on many of these points, but I guess, you know, when we sort of, you know, got into this, our main motivation for doing it, and it's like, okay, why are we in this business if there are so many tools that are doing this already? So one was this black box factor that I already mentioned to you. The other, we felt that there was really nothing to give us the ability to integrate multidimensional data, and by this I mean several sort of, you know, first of all, like, you know, metabolomics data, for example, and gene expression data, and, you know, potentially proteomics data. The other is sort of variety of sort of, you know, time points and sort of, you know, I mean time dimension. Right, and then we were really sort of, you know, not satisfied with relatively narrow view of metabolic pathways, where you can sort of, you know, look at one pathway at the time, and we wanted to take more of a network-based approach. And I think after the break, I'll talk a little bit more about sort of, you know, linking metabolites to diseases. Okay. And so this is an overview of our tool, Netscape, and you have seen the earlier version of this slide. So this part here really refers to pathway analysis, and after the break, I'll talk to the brand new correlation module, and you're going to see a demo and do sort of, you know, this part hands-on and see a live demo of this part. But so the workflow is really pretty simple. You can start here with gene expression data and metabolomics data. I just have to mention to you that, I mean, you can actually put proteomics data in, and in fact, if you have SNPs, and, you know, as long as you have entree gene ID, and, you know, have a list of genes that these SNPs sort of, you know, belong to, you can input that type of data as well. And so another optional input to the program is a list of enriched pathways or any other biological categories. And George sort of, you know, gave a nice overview of the enrichment analysis method. So if you have your enriched pathways, in particular from gene expression data, you can sort of, you know, provide that as an input. And what the program will do, it will sort of take this input data and it will attempt to build the metabolic networks incorporating the data that you provided. And so how do we do that? This essentially right there in the middle of the slide, it shows the sort of, you know, main objects in our database. I mean, there is a database behind this tool. What I neglected to mention, but I think it was said earlier, that Netscape is essentially a plugin for an open source software that is called Cytoscape. Cytoscape is a platform for network-based visualization. And it's not specific. I mean, you can use it to visualize any network you want. It can be electrical grid, it can be a social network, or it can be a biological network. And what Netscape does, it essentially takes this Cytoscape framework and customizes it specifically to build and you know, visualizes metabolic networks. And there is a database behind it that combines data from these pathway databases. And I will tell you where we're sort of, you know, going with that and future directions in terms of, like, you know, consolidating data from different databases. But essentially, these are the main objects in our database. We have compounds, we have reactions, and a compound can be a substrate or a product or cofactor. And we have information about enzymes that catalyze the reactions and genes that encode enzymes. Which is why I said that, you know, as long as you have essentially as long as you have gene ID, untrade gene ID, for example, or gene symbol, you can map that information. If you have protein data, but if you can map the, I mean, hopefully you have, you can map this to genes, then you still can input proteomics data. And I just want to stop here for a second and introduce Bill Duran, who is the main developer of Netscape. He's sitting right there on the back, and Bill will lead the demo tomorrow. And the group of people here, Terry, Tim, and Glenn, have been involved in developing Netscape 2. And Marcy Brandenburg will be also leading and helping with the demo tomorrow. And we really are indebted to her, because all the instructional materials, we have video tutorial and we have very nice user manuals. Marcy created all of these. So we all of that to her. And of course the it was sort of, Chuck inspired the development of this program. He came to, we talked one day and he said, well, it would be really nice if I could do this. And so, he can tell you how close we have come to doing this, or, I mean, or do we still have ways to go? But nonetheless, let me sort of go to this example that has been mentioned several times throughout the workshop, through this study, because this is what we will be using for the Netscape hands-on session tomorrow. This is from Chuck's lab, actually, where they looked at the beta cells, and so beta cells which were pre-incubated with either glucose or high concentration of fatty acids. And so I understand that they looked at the effects of these pre-treatments on the sort of metabolic pathways after the cells have been stimulated with glucose. And this heat map essentially gives you an overview by different classes of metabolites, what is happening in these cells over time. So there are different sort of, groups and classes of metabolites. So the data sets that you will be using for the demo tomorrow essentially is derived based on this, from this sort of, experimental data set, and based on this publication. All right. So if, so you will see, those of you who will attend that, you will see how we can sort of, take this data, and we'll show you how to load it into Netscape. But this is sort of a preview what you're going to see. So one thing about Netscape is that it allows, so it can handle pretty large networks. As you can see, and I mean, this particular network here resembles a hairball. Okay, and so we don't really restrict the size of the network that you can create. It's only limited by the, essentially, I think, memory and RAM on your computer. But what we decided to do instead, we decided that, it was sort of a design decision, if you will. We decided that we will sort of invest our efforts into various filtering mechanisms. And what you see down here, even if you cannot read it, is essentially a list of pathways that, involve the compounds that you see in this network. Okay, so I don't have time to go over the details of this graph. But I just want to point out to you that it's very easy with a click of this button. What you can do is you basically can select a pathway, and I have TCA cycle selected here just as an example, and you can create subnetwork, and it instantly becomes much more manageable. And there are various visual features to show the metabolites that are up and down regulated. Similarly, you can load genes and visualize these. And so I mentioned to you that this particular study is a time course, and you're going to see tomorrow how you can sort of view these changes over time. And another useful feature, so I told you earlier that we did not want to be limited to a particular pathway. In Netscape, there is a very easy way. So here, basically, you can right-click on a compound. This pink node here is oxaloacetate, and it's unstable, so it could not be measured directly. But if you wanted to sort of, bring more compounds in the network, you can right-click on it and, go through this set of menus, and it will allow you to expand the network right there. So you can basically, walk the pathways and, like, different directions as you see fit. And, as you see, what is highlighted here, a few new compounds that were brought into this. Again, you're going to see it in a lot more details tomorrow, so I'm not going to stop there. But another feature of Netscape is that it allows you to do this side-by-side displays. This is the same network, and so the first condition here is control. Second is free fatty acids and high glucose concentrations. And so this little window here allows you to control these changes over time. So again, I'm sort of, moving to the last slide here, and because, I think we're running behind. So I hope you will get a chance to try it and see. We tried to make it user-friendly. It's meant for the sort of someone who does not have any sort of, special programming skills, and it's meant to be user-friendly. I talked to you a little bit about dynamic visualization and, static assessment, about inputs. And so the things that I'm going to mention are sort of, I'm going to show you some of the sort of, links to additional information that you can get from Netscape. You can get more information about different sort of, if you click on any node, you can, get information about compounds, including links to various databases that I've mentioned, compound structures, and, various other details. So I think I'll just stop here. We can have a break, and when we resume, I'll talk to you about some other tools that we've been developing. Any questions at this point? Yes?
Two questions. First, if you have like a feature, all the possible cycles, and you pick one metabolite and you can see it's being produced in this cycle and being used in alternative, just to visualize, instead of using all the biochemistry book where your metabolite can go.
Yes, you can. Assuming, the only thing that, well, when you said feature, that sort of implied to me that it was maybe an unknown metabolite, okay? So, it has to be a known compound, okay? And it has to be a known compound, and we have to be able to map it. And I will talk to you after the break about some difficulties in doing this mapping, because, these are, like, apart from sort of, methodological problems that George talked about, there are some very sort of, practical issues that sort of, limit our ability to do that. So we'll talk about that. Yes, that's kind of the idea. And the second?
And based on your experience, how much correlation you have between genomics data and metabolomics data?
It's a very good question. I think it's very hard to sort of, establish direct correlations, okay? So there are several issues. Again, I'm going to talk a little bit about that. I think the things to consider is, how much correlation should you really expect? Are you doing tissue metabolomics, and are you doing gene expression in the same tissue? That's the first question to ask. And this is not always possible, right? It's not always the case, right? If you're doing plasma metabolomics and gene expression in tissue, how well would this correlate, okay? Then there is another factor to consider is really the timing, right? So the thing to keep in mind is that at least some changes in metabolome, they're very rapid. And there can be very sort of, rapid response. And in gene expression, there are changes over time, but they're not as instantaneous a lot of times. So, there are a lot of, there is really, I don't know, I think the short answer is not a lot of correlation. But, we see some. I don't know how to measure it, I guess. There are some good publications out there. It's a little bit easier to, if you're asking a question, how much correlation is there, maybe it's easier if you look, for example, at yeast, where there is like one cell, and even there is not a whole lot of direct correlations based on what I've seen and read. In human, or it's even more difficult. Is that?
It's always more difficult.
Yeah, can go wrong. Yeah, saying that. Yeah.
Based on this correlation of genomes, proteomes, and metabolomes, is it possible to predict some unknown metabolite based on this, or reverse?
Is it possible to? Well, we haven't tried predicting metabolites, or I haven't tried predicting metabolites from gene expression of proteomics data. I suppose if you have a targeted study and you know that there is like a lot of enzymatic activity, you can look at specific metabolites and get good results. What we are trying to do, and what I'm going to talk about, is we are trying to predict the unknown features from metabolomics data. Okay, by looking at correlations between metabolites, because the sort of idea is essentially guilt by association, that compounds that are co-regulated, or otherwise linked, they would change in a correlated fashion, and that's what I'm going to talk to you about. Okay, I think that genome and proteome just adds more complexity to it, and we don't really know. All right, thanks. I think we have a break.